Oh, sorry. How are you?
All right, everybody, we'll go ahead and get started. Good afternoon. Obviously, I'm not Gavin. I'm going to be doing the Good News Thursday. To start off, the exercise medicine on campus received the gold status from the ACSM, and it's the first time ever. In addition to that, Alexa actually received the ACSM Leadership and Diversity Training Program and will be receiving full funding for traveling to the national conference in May. So James told me yesterday that he was actually set to finish his participant data collection this week for his dissertation. So
That's a
and then finally, Dr. Elizabeth Park has been selected to be the keynote speaker at Precision Nutrition Symposium at Arizona University. Congratulations. I'll go ahead and hand it over to Deputy Introduce our speaker, and I'll just send the sign up sheet back.
Thank you very much. It is my pleasure today to introduce our speaker, Dr. David Baranger. Dr. Baranger is currently a senior postdoctoral research associate at Washington University in St. Louis in the Department of Psychological and Brain Sciences. It's also where he completed his Ph.D. Prior to that, he interned at Duke University and was a research assistant at NIH Institute of Mental Health. He's been extremely productive and currently has 32 peer-reviewed papers in top journals, including JAMA Pediatrics. He's currently funded on an NIH K99 R00 grant with NIAAA entitled Reciprocal Longitudinal Associations Between Brain Function and Alcohol Use Trajectories in Adolescents and Young Adults. Today, he'll share some of his recent work in this area, and I know we're all excited to hear about it. So please join me in welcoming Dr. Baranger.
Well, thanks very much. Thanks for the kind introduction. Very happy to be here. This has been a really lovely visit. I really enjoyed meeting everybody. Students over lunch. So I'm David. I'm a postdoc at WashU. I work with Ryan Bogdan and Arpan Agrawal at WashU. And I'll be talking about work, line of work that I started during my PhD, and that's kind of continued throughout up into this time now. And I'll talk also about some of the future studies I have planned in the future. Let's see how to advance. Okay, like that. So broadly, I'm a neuroscientist. My PhD is in neuroscience. I'm interested in the brain. I'm interested in how our brains influence decisions around using substances, around when we initiate, when use accelerates. I'm also interested in the converse, in how substance use in turn influences the ways our brain develop and function. I have interest as well in a variety of moderating factors, genetic risk factors, environmental risk factors, which I'm gonna, have image of a person who's pregnant, preludes of my work looking at prenatal risk factors. And I take two approaches to this question. One is a big population data approach, samples of thousands and tens of thousands of people, and then I complement that with targeted focus studies, which mean smaller number of subjects, and I kind of view these as two complementary ways of addressing these questions. So not everyone here studies addiction or even alcohol, so why care about this question? So one reason is that binge drinking, so heavy drinking, so 4 drinks in a sitting for women or 5 drinks in a sitting for men is extremely common in the continental US. Up to 22% of adults reported in a survey in 2020 that they had binged at least once in the past month. And for these binging adults, this can be many more than 5 drinks. I think about a quarter of binging adults report over 9 drinks in a session. So very, very heavy alcohol use, and it's very, very prevalent. And I mean, I probably don't have to tell you this, but it's a major health problem. It's estimated that up to 30% of deaths can be attributable to alcohol use across the US. So this is a major health concern, I think, for all of us. And of course, it's also a costly health concern. You can estimate that on average, each drink consumed has cost society about $2. In terms of healthcare, productivity, and crime and mortality, or overall, about 250 billion in the year 2010, and that's only gone up since. So alcohol use is a big problem. Something that I find quite interesting about alcohol and probably mental health disorders is that they're really disorders of young adults. So the peak age for risk for the onset of alcohol use disorder is only 19. And by the age of 25, it's estimated that a full half of cases will have onset, and this is worldwide data. This isn't US specific. So I really view substance use as a developmental problem. It's a problem that clearly onsets quite young for the legal drinking age in the US. And this period of time coincides with the rapid brain development that happens during the same period during adolescence. So I think this really has influenced how I approach this question. I use a lot of developmental samples. I use a lot of longitudinal data, and a lot of my work kind of takes a more developmental lens. So now, we're talking about addiction in the brain. There are kind of two models to keep in mind. One is a model that's referred to as the stage-based model, which is a model that talks about the effects of alcohol and potential causal effects of alcohol in the brain and how that has behavioral consequences. Starting with binge drinking. So we all know what binge drinking is, and that is kind of the rewarding aspect of alcohol that relates to the brain's reward system, which is called the basal ganglia, or sometimes the striatum. That eventually feeds into this negative affect component where there's kind of a blunted mood that results from extended kind of unreasonable rewards, you might say, from alcohol. So anhedonia, clinically we call, so kind of depressed mood that relates to amygdala circuits, the hippocampal circuits down here in red. Eventually, one becomes preoccupied with obtaining the substance, anticipating the substance and working towards obtaining the substance as a reward in its own right. And that, of course, is going to be governed by circuits that drive cognitive behavior and cognitive control at the prefrontal cortex. So I know none of you are neuroscientists, so don't worry about it too much, but the basic idea. And then the second model is a model that relates to risk. And this is basically about a model about why adolescence is the period when we're at highest risk for experiencing the onset of a substance use problem, which is that the trajectories of development of this prefrontal cortex and the striatum, this reward region, are different. They're not parallel. The striatum develops faster than the prefrontal cortex, and that drives accelerated increased reward-seeking behavior in adolescents, which we consider to be normative. Adolescents are dumb. They take mistakes. They don't wear their helmets. They go out and they do things that no reasonable adult would ever consider doing. And that is part of being a kid. It also makes you at increased risk for substance use problems. So I think both these are important to keep in mind. So the question that started off this line of train of thought was a simple one, which was it's very easy to observe that alcohol is correlated with having a lower brain volume. People who have alcohol use disorder have smaller brains, and that is the most replicated neuropsychiatric finding you will find in the imaging literature. And people are always going to tell you, well, alcohol kills brain cells. It's going to be a causal effect, right? Actually, it's really hard to know if that's the case. And very little attention had been paid to this alternative hypothesis that actually maybe there's a third variable at play. Something like genetic risk or environmental risk factors could be driving both of these measurements, causing them to be correlated. I think this is also important, an important thing to keep in mind, regardless of the research question, just in terms of research methodology. How can you take a correlational observation and start to make causal inferences about the underlying mechanisms? So as a first stage, I just wanted to identify replicable correlations between alcohol use and brain volume. So I took two samples. One was a sample of college students from Duke University. The second was a sample of adults from Missouri collected around St. Louis called the Human Connectome Project. And we found two regions of the brain, the insula. So this is a cognitive control region. It's involved in kind of integrating information across multiple regions, and the this is we call it the superior middle frontal cortex, which is another way of saying the prefrontal cortex, which is that region I had up a couple of slides ago. Both of these are smaller when you drink more. And this is a measure, this isn't alcohol use disorder, this is just moderate alcohol use. Okay, what can we do to infer whether there's a causal effect or not? So one strategy is we can use twins. I asked this, I think, to the graduate students, but twins are amazing, an amazing population. Twins are an amazing population because they have the same, or very similar, if you're using dizygotic twins, genetics and environmental background. So you can control for all of these factors that might be plausibly contributing to your measurements. So here's kind of what you might do. This is what's called a dichotomous analysis. We take our siblings or twins. Some of them are high drinkers, some of them are low drinkers, and then we have different kinds of twin pairs. Some twins are the same, they're concordant. So they both drink high or they both drink low. Some of them are discordant. And then one of them's a high drinker and one of them's a low drinker. And we're going to compare all these different groups. So in all cases, we're going to expect that our low drinkers, or both siblings are low drinkers, we're going to expect them to have greater brain volume than the pairs where both siblings are high drinkers. So this is our correlation just repeated. Key comparison, and this is a hypothetical model, this isn't the real results. The key comparison is what happens with these discordant siblings. So they're matched for genes, they're matched for the environment, but they drink different amounts of alcohol. If they differ in their brain volume, that gives a signal that there might be a causal effect of alcohol on the brain. If they don't differ, there's a couple of different potential outcomes, but these will all be interpreted as reflecting the influence of pre-existing risk factors. We call one the predispositional model and one the graded liability model. In both cases, we expect these siblings who differ in their alcohol use to not differ in brain structure. And that tells us that there's something else going on in this correlation that isn't a causal effect. We turn the data, we of course see our expected greater volume in the low drinkers versus the high drinkers. You look at the discordant siblings, a lot of variety in the data, variation, but the key observation here is that these sibling pairs don't differ in the frontal cortex, and they also don't differ in the insula. So that tells us that this correlation we're seeing maybe actually isn't a causal effect at all. It's actually, there's some sort of pre-existing risk factor. Okay, we think about, well, can we probe that more? Can we push it a little bit more? So one way we can push it is we can say, well, if there's a pre-existing risk factor at play, then brain structure should be predictive of future alcohol use. And we can show that's the case. So this is a study, a longitudinal study of teens. This was collected in Texas, who were non-drinkers at baseline when they were scanned. And then they were followed longitudinally for several years, and a bunch of them started drinking. And we can see that brain volume is predictive of a later age of drinking. So those who have the highest brain volume, so brain's biggest, they tend to onset their drinking at a slightly later age. So that indicates that there's some sort of longitudinal effect. That means that causal effect of alcohol on the brain couldn't explain this pattern of data. Okay. Now, what could be the causal risk factor? Maybe genetics. Genetics is an easy place to look. So we did some bioinformatics. So this is all publicly available data that I'm talking about now. This is data of postmortem whole cell RNA-seq, collected as part of the GTEC project. I have a bunch of brain regions, and I also added in the liver just for completeness. And what we're testing here is whether genetic risk for alcohol use, and that's assessed using an independent study that looked at genes that are correlated with alcohol use. Is genetic risk for alcohol use correlated with mRNA expression in these post-mortem samples in the brain or in the liver? Right. We see a handful of signals in the liver, nothing very robust. We do see one signal in the frontal cortex, the same brain region that I've been talking about, that survives our much more stringent Bonferroni correction, and that signal then replicated in a fully independent set of analyses. It's So that points to the plausibility of there being a genetic effect. And more recent studies have taken other methods and have again seen that there are genetic influences that potentially drive these correlations. So this was kind of a first pass, and we've been pursuing this line of inquiry along a couple different ways. Most recently, we've turned to some really big data studies that were led by my colleague Alex Miller, who was previously here at Mizzou. And we use a data set called the ABCD. So this is a study of 11,000 kids collected at 21 sites across the US. And then they were followed longitudinally. Now they've been followed for 5 years, and I think they're going to be followed for another 5. They're scanned every other year. There are a bunch of other measurements, physiological measurements, behavioral measurements, cognitive measurements. And we can see that they start using drugs. A bunch of them are using at the baseline scan. And here we're defining use as any kind of initiation. So any non-religious sipping of alcohol counts as use here. And, taking a puff of a joint, a puff of a cigarette, borrowing your mom's e-cig, all of those count as use initiation in this context. We see that there's a lot of low use of cannabis and nicotine early on, and then there's a kind of a nice increase as they get older. But of course, most kids are starting with alcohol, and most of this nicotine use is e-cig use. Very few of them are smoking cigarettes. And so we can ask, okay, is brain structure correlated with future initiation in this now enormous population sample? One thing that came out that we thought was pretty cool was that actually it is, but not in the way we expected. We actually see that kids who go on to use or are currently using substances, particularly alcohol, and there's also, so here, I'll orient you a little bit, there are different brain regions here. The y-axis, these are actually whole brain measurements. So the whole volume of your brain, the whole volume of just your cortex, which is a part of your brain. We also can talk about the thickness of your brain. Your brain is you can really think of your brain as a balloon that's been scrunched up. So there's a thickness to the balloon, and there's also a surface area to the balloon. So we can talk about the thickness of the brain, surface of the brain. Your brain is obviously folded and looks all weird and mushy, so those are called sulci, and they have a depth. So you talk about the depth of those. And we see is actually that the kids who go on to use actually just started out with bigger brains to begin with, which is quite interesting. Kind of goes a little bit in contrast to what I was just talking about, but does point to actually it isn't necessarily like a small equals bad story, but it's a more complex story. And this actually, we think, points to socioeconomic risk factors, that kids who have early onset of drug use tend to come from better off families, typically those who start using alcohol early. And so we're actually seeing like a wealth effect in these data. We look at the individual regions. So again, a bunch of brains, different brain regions, looking at volume, thickness, surface area, depth. Again, black regions are FDR significant, gold regions are Bonferroni significant. A bunch of the same regions that I was just talking about, the frontal cortex, these are again showing up as being smaller in kids who go on to use substances, any substance, and particularly alcohol. But I think quite interestingly, we're actually seeing increases in the back of the brain, in these regions that are important for visual processing and regions that are important for motor control. And so this, we think, actually points to not necessarily like a simple, like alcohol equals small brain, alcohol equals big brain, but actually the axis of the brain, you can think about there being like a back to front axis to your brain. And that kind of strength of that axis is very tightly developmentally regulated. And we think that what might actually be happening is that some risk factors for substance use may kind of disrupt that axis in terms of the cortical patterning and differentiation early in development. Of course, I mentioned before that a bunch of these kids were using substances to begin with. So if we actually just look at only the kids who were not using substances when they were first scanned, we see a very similar pattern of association. So here on the x-axis is the effects I just showed you. They're just in a scatter plot now. And on the y-axis are the effects if we excluded everyone who was using at baseline. And these correlations are really tight and really positive and very significant. So whether it really looks like it's an effect of pre-existing risk, not an effect of onset of use or like earlier use. So more broadly, this is from a review we recently published. More broadly, and this is summarizing across not only our studies, but now the literature has really exploded. There's a lot of evidence that these gray matter correlates of alcohol use and drug use really reflect what we call predispositional risk, pre-existing risk, potentially genetic, potentially environmental. And there's a lot of convergent evidence across studies and methodologies that point to effects in the frontal cortex, the whole brain effects, in the back of the brain, regions that are important for motor control, like the cerebellum. So I think this really changes how we think about drug use and how we think about brain, and thinking about how we need to work on disentangling these factors. And in the future, we're going to start doing studies using these big data sets to actually ask, are the trajectories of brain development different in people with differing levels of risk? Not just measuring the brain at one time point, but let's actually look at the slope of development and ask whether that differs depending on risk. So this kind of spawned an interest in understanding risk factors. And one risk factor that I became interested in just because of current events is prenatal cannabis exposure. So obviously cannabis use is increasing dramatically across the US. Between 2002 and 2016, it almost doubled. And that was paralleled by an almost doubling in use by women who are currently pregnant. So more and more people are using cannabis when they're pregnant. And a lot of that is due to use when they're in their first trimester. And this is concerning because we have evidence both from preclinical models and from patient studies that prenatal cannabis use is associated with lower birth weights, it's associated with premature birth, it's associated with increased rates of NICU admissions. Cannabinoids can cross into the placenta, they can cross into the brain, the fetal brain, and receptors are expressed throughout the placenta and throughout the brain. So there's a lot of evidence to suggest that this might actually be having effects on brain development. Some work that we've done to suggest that might be the case, again, has come from the ABCD study, where we've seen that kids whose mothers used cannabis before they knew they were pregnant and continued to use cannabis after they learned they were pregnant had worse mental health across the board. So the total problems were worse, rule-breaking behavior, they had more psychotic-like experiences, ADHD was worse, aggressive behavior was worse. So it suggests a potential effect of prenatal cannabis exposure on the brain. And interestingly, again, in the study led by Alex, we looked at these predictors of early cannabis use initiation in particular, and we saw that prenatal cannabis use was the strongest predictor of early cannabis use initiation, stronger than factors like impulsivity and even stronger than factors like self-reported intent to use cannabis previously. So our question was, does the brain mediate these effects? We took a very exploratory approach. So here we're showing, so y-axis is the p-value, it's on the log scale. This dashed line here indicates that FTR corrections for all regions. And there are a lot of measurements we can generate from imaging data. We can look at these structural measures I was talking about, the volume and the thickness and the depth. We can also do what's called a resting state scan, where someone is laid in the scanner and they do nothing. And we can look at the flow of oxygenated blood in their brain. And that's a proxy for neuronal activity. Because neurons, after they activate, they will eventually ask for more resources that causes there to be influx in oxygenated blood to that region. And that is a way for measuring brain function and brain connectivity. We also can do what are called white matter scans or diffusion scans. So water diffuses in the brain, and it diffuses differently in the brain in different tissues, different rates, at different speeds, in different directions. And we can use various models to try and figure out what kind of tissue are we seeing? And we can like, okay, let's look at white matter, look, axons and then brain. Okay, how what's the integrity of those axons? And we can ask, okay, what about the neurons and the gray matter? What about the extracellular diffusion, intracellular diffusion? We can tease all this apart using white matter imaging. And it tells us different information about inflammation, about synaptic pruning, growth. So I'll talk about two of the regions that we saw kind of the strongest signal for. So one is this region, it's called the pars triangularis. Thank you, Latin. It's the front right part of the brain, and it's in the brain that's the region that's important for language and cognitive control. And we're looking here at a measure that is a measure of diffusivity through the gray matter of this region, and which is lower in people who've been exposed to penile cannabis. And that potentially indicates a kind of inflammatory effect at play here, or also potentially, it's also consistent with an aging, like an accelerated aging interpretation. And the second observation we saw was for a tract called the forceps minor. This is one of the key tracts that connects the two lobes, the two hemispheres of your brain, in the frontal part of your brain. The part of the corpus callosum, we call it, and this was increased in people with penile cannabis exposure. Again, consistent with an inflammatory interpretation, or potentially like a disrupted early pruning interpretation. It's kind of hard to know, what do you make of these regions? I don't really know. So we went back and asked, okay, are these regions correlated with mental health? And they are. They're both correlated with what we call externalizing psychopathology, so ADHD problems and attention problems. So that actually points to a potential, mediating effect, that cannabis exposure could have an effect on psychopathology via changes to these brain regions, which is all pretty consistent. And we can run a longitudinal mediational model, and we see that yes, indeed, the mediational model is significant. It's very small, of course, in both regions, but it's certainly present that these effects might be mediated by brain, potentially by brain development. We're not entirely sure yet. So we can say that penile cannabis exposure is associated with some localized brain differences. We think the mediation effect is plausible, and it could reflect reduced inflammation or accelerated aging of some sort. The main caveat, of course, is that this only explains a fraction of the proportion of the risk for adolescent psychopathology, which is always what happens when you have this kind of data that's really big and extremely kind of low depth in terms of the behavioral phenotyping. They're always going to see small effect sizes. And I think it's also consistent with the fact that we're talking about environmental exposures that occurred 10 years ago. So you're never going to see a huge effect if something happened 10 years down the road. I think broadly, these are consistent with healthcare recommendations to abstain from cannabis use when you're pregnant, though I would not say that this data in of itself is enough to warrant not using cannabis when one is pregnant. Of course, people use cannabis when they're pregnant for all sorts of reasons. They're experiencing extreme nausea, they're dealing with other sorts of mental health problems, they're concerned about using SSRIs when they're pregnant. So there are a lot of reasons to use cannabis when you're pregnant, but I think it supports the idea that one should be cautious surrounding that decision. So I've talked about this link, this kind of like brain to behavior link, and in the future I'm going to kind of try and connect the dots, go the other way. First, I'm going to do that with mental health. So we've done some studies where we've tried to ask about bidirectional associations between drinking and mental health. So this is a study that's led by a really talented grad student in the lab, Sarah Paul, and this is using a longitudinal, again, multi-site study called KOGA, where we have a bunch of participants who come from high-risk families, and they were followed longitudinally over many, many years. And so we have data on diagnoses for alcohol use disorder, for when they first started drinking. We also have data on symptoms for depressive episodes, for anxiety, panic symptoms, suicidal ideation, ADHD, all sorts of mental health traits. And we see, again, our expected kind of effect that alcohol use it predicts future ADHD symptoms as expected. It predicts future changes in externalizing. This is kind of an effect that we've seen again and again. Oh wait, that's backwards. Externalizing predicts future alcohol, which we've seen again and again. And we've known this for a long time. It's nice to see that, that we can then basically reproduce that result, but we can actually do the converse. Does alcohol predict changes in future behavior? Of course, ADHD has such an early age of onset, we couldn't ask whether alcohol caused changes in ADHD longitudinally, but we do see evidence that alcohol, first drink and first diagnosis of alcohol use disorder is associated with increases in depressed mood, increases in suicidal ideation, and lots of increases in conduct problems. So we're seeing evidence of this kind of bi-directional effect for both some particular externalizing, what we call externalizing traits. So things like ADHD and conduct disorder both are causes and consequences of alcohol use. We've also, so this is with longitudinal data. Another way you can ask this sort of question is with genetic data. So this is an analysis where we use just the summary statistics that are produced by genome-wide association studies. So you can do this analysis with anything. You can do it with, here I'm doing it, we did it with cannabis use, tobacco use, and schizophrenia. You could do it with anything that a GWAS has been done on. And you can ask whether the patterns of associations are consistent with different kinds of causal models as a way of getting at causality. The question about whether cannabis use causes schizophrenia or whether schizophrenia leads to cannabis use has been an ongoing one that's been debated in the epidemiological literature for a really long time. And what we see is actually using this kind of causal modeling, the answer is yes, both are true. So we can see that cannabis use at the bottom here is causally associated with increases in schizophrenia risk, but schizophrenia risk also is causally associated with increases in cannabis use disorder. So again, we think these bidirectional effects occur a lot. There really isn't potentially really such a thing as a unidirectional effect, at least when we're talking about mental health in the brain. A lot of these things kind of go back and forth, and there's a lot of complex interplay between them. So that leads me, to my future directions, which is going to be trying to zoom in on a particular single piece of the drinking puzzle, which is zooming in on this binge drinking piece right here. And thinking about, okay, well, can we untangle this a bit more? A lot of the data for our models of how binge drinking influences the brain come from preclinical data, and preclinical data is fantastic for a lot of things. We really love preclinical data, but data from rodents don't always translate, and it's really nice to be able to show that we might see the same effects in humans. The kind of effect we might expect to see would be something where, binge drinking has an effect on the reward system. This is just an image from a meta-analysis of the reward system, but you would also expect the reward system to then influence binge drinking. So binging, causes an increase in reward, okay, that we learn, we pair binge drinking with reward over time. Then when we're feeling like getting rewarded again, we're going to go look to alcohol for a future reward. But of course, this developmental model would suggest that there might be some kind of moderating effects of this kind of prefrontal cortical cognitive control circuit, where you'd expect people who have better cognitive control to be less susceptible to this kind of feed-forward loop. So you'd expect some sort of moderation in this pathway. And so the goal is to probe this a bit further. So what we're going to do is we're recruiting college kids who binge intermittently. So this is data from WashU. So data from Mizzou would be a lot worse, I'm told, I think with authority. But at WashU, about 35 undergrads have a score on this drinking questionnaire that we would consider to be indicative of hazardous drinking or indicative of potential likely dependence. This is a high amount, and I think probably would be even worse here. And of these kids, 44% of them are binging at least once in the past month, if not more. But this actually presents a really interesting kind of natural experiment. College is a really big risk factor for drinking, and kids in college kind of behave in this very stereotyped way where they drink only occasionally, and when they drink, they tend to drink quite a lot. So what we can do is we can plan a study around that behavior, where we ask them to report, like, okay, when are you planning on drinking? When's the frat party? When's spring fling? When is your friend's 21st birthday? Whatever the events are, put them down on a calendar, right? And plan a bunch of fMRI sessions around their schedule. So we're going to scan them, right after, a day or two after they've been binging. And we're also going to scan them, during exams or some period when they've got a midterm and they haven't been drinking as much. So you got a lot of within subject behavioral variability in the drinking behavior. And quite uniquely for an imaging study, we have 10 scans over a period of a couple of months, which is really never done in imaging. Yes.
Your scans, what kind of standards do you have for them? They have to be like fasted or?
None of that. None of that. Yeah, because we're looking at the oxygenated blood flow, which has, and we're really interested in normative oxygenated blood flow. So it's more like, I want you to come in your natural state. I'm not going to do any kind of like fasting or don't drink water or make sure you have a meal. Like I just want whatever they do. I want the natural condition. Any time of day. And honestly, with the way scheduling works, it's usually at night. In college kids' schedules, it's usually at night. Yeah. I mean, which is, I think so. There are some concerns in terms of like, oh, well, they're going to be tired, so you want to try and control for that and have some morning scans, or you try and just record like, oh, did you have a caffeine drink, or like try not, I might be like, try not to, have a monster before you come in, but some of them will, and so you just ask them. And we're going to collect what's called EMA data, so ecological momentary assessment. So this is a way of data collection where you give people daily smartphone questionnaires. And you ask them about like, okay, did you drink today? How's your mood? How well did you sleep? And we're just going to collect it every single day, so we have some information on their actual drinking patterns. And if someone indicates like, oh, actually, I've been drinking a lot, you might actually say, well, actually, you want to come in sooner? I have a scan available tomorrow. Like, let's reschedule this to get you in so we can get a better signal out of this. And as I was saying, this approach of collecting 10 scans over a short period of time is usually not done because a lot of the emphasis in my world is on between subject effects. So you would probably instead rather have 300 people who come in once. But what this lets us do is it lets us really get at the within subject effects, because we have a lot of data per person in various states. And so we'll get, tasks that are related to these cognitive control constructs that I was talking about, a task related to reward behavior that I was talking about, as well as the scans that measure the things that I was saying, thickness and volume and surface area and all of that. And all of that will be measured throughout. That's the ROO, that's going to start happening soon. And then in the future, some of the future studies we're going to do are going to be focused on things like cannabis use, so substance use. So a big question in the substance use literature is, are the effects we're seeing addiction effects, or are they alcohol metabolite effects? And you really need to know whether it's one or the other. So one really great way of doing that is being like, okay, let's get people who use cannabis in, let's get people who are using some other party drug in, and we can start kind of comparing and contrasting across substances and looking for agreement across the different substances and those of use. Another one is going to be looking at kind of stages of substance use. So, one idea is to find people who have been abstaining, but maybe plan on initiating your use around their 21st birthday, which is like not actually that uncommon. So we can then get people who are like alcohol abstainers, and then we can get heavy alcohol use at the 21st birthday and subsequent increases in alcohol use as another nice natural experiment. And finally, we'll start, I'm planning on starting some collaborations with people who are looking at treatment that start thinking about, okay, can we take some of our findings and think about how we could potentially move them into the clinic in different ways. And in addition to all this, this focus data analysis will also be in kind of in tandem doing a lot of these big data analyses with the ABCD study and other data sets that are out there. And with that, is thank my colleagues, mentors, and collaborators, and all of my funding. And I'm happy to take any other questions.
About dry January.
Yeah.
Question, very nice talk.
Yeah.
Have you considered doing more control studies in which you subject subjects to given amount of drinking and do scans before and after? You could even do that intermittently.
Yeah, totally. Yeah.
Some of the noise that you might have otherwise.
Yeah, absolutely. Absolutely. I guess, my observation with imaging is that the most reliable effects are the really huge ones with big, when you have a lot of alcohol in your system. And I've been very worried that at like lower doses, we might not have the power to see effects. And
So are you afraid that IRB would not approve a higher dose?
yes.
Can you comment on this? Because you've done studies.
Yeah, well, they did approve it,
Yeah.
awful lot of drinking, but
Yeah.
I think that the uniqueness of what he's doing is that he's after the daily noise. After, nobody is collecting.
No, it's a very different way.
I mean,
We usually try to get away from this.
yeah.
Yeah.
You kind of want.
And think about how we stated that will help other people in the future figure out about effect size or normal changes, or does this dose even matter if the normal noise is, it's really hard to get these kinds of data. Do you think this intentional, sobriety of dry January that just overtook the United States with a lot of people?
Yeah. That's a really cool idea. I mean, the hard part is that scan time is competitive.
Yeah, right.
Um,
Everybody has to be in it.
So it'd be the kind of study where you'd be like, I am gonna like kill my RAs for 3 months to
Yeah.
get this data. But it'd be super cool, especially if you could do like scan in December, get the month prior, get January and get the month after. That'd be a super cool study. It'd be really hard to
And
run.
I just read an article that this year, the dryness extended for almost 3 months. Many.
Oh,
people
cool.
said, I lost 10 pounds in January, I'm gonna keep going.
Nice.
And so now the effect of dry January
Yeah.
is starting to clear off.
I mean, I think there are lots of cool potential interventions in terms of like also maybe providing people with non-alcoholic beer. Like you can imagine, like I'm just gonna give you a non-alcoholic beer. I'm not gonna tell you what to do with it. I'm just gonna give it to you, and then we can see how that changes their behavior and how that changes brain function. As kind of a, kind of maybe the opposite of inducing alcohol use, but like reducing it. Yeah. I'm not good at this. So we start around the room and go left.
Good. Thanks for your talk. I'm curious if with your measurements of the brain in the younger cohort, essentially, if there's any sort of normalization that goes into that, in the younger age groups, everything's 5 percentile. So, do you measure their head circumference and say, oh, well, this one has a bigger brain, but I can see their head is this size. So
Yeah. So there's a lot of, oh my God, so much normalization happens. So everyone's brain gets remapped to a common atlas. So that gives a common space in order to think about the brain and its shape and where we are. And there's a variety of algorithms that I cannot tell you about that do that. Once you have that, then you usually kind of will correct for what we call the intracranial volume. So the head size has a covariate in your model. And then you look at like the subregions below that. Because exactly. People with bigger brains have bigger skulls, tend to have bigger brains, and that isn't necessarily actually very interesting. So we're trying to remove that. That's kind of the main kind of normalization that happens, but there are several other steps in between. Yeah. Let's see.
So outside of academia, right?
Yeah.
Like,
Yeah.
it is very common that trauma is the root cause of it,
Yeah.
right? But I kind of have yet to hear anybody talk about like ACE scores being one of the
Yeah.
factors, right? That predicts onset, of use.
Yeah.
Or disorder development or any of that. So
Yeah.
I just kind of wanted to know your.
Yeah. So I think There's a couple of tricky pieces here. ACE scores are correlated with AUD for sure. So a couple of hard parts are like, there's a lot of disagreement about what ACE scores are measuring. So when you talk about childhood trauma, what are you talking about? Are you talking about the actual event? Or are you talking about how the person experienced the event? And actually, most of the data points to the importance of childhood trauma being how the individual interpreted the event and how they experienced it for themselves. Because you can have people who experience, 5 soldiers going to the same battle, 2 of them have PTSD. Why is that? Because of their subjective experience of the events that occurred to them. And then that gets into the really tricky business of trying to figure out how to measure subjective experiences of an event that occurred 10 years ago and normalize it across people. So I've done some work in the trauma and early life stress space. Very hard, a lot of disagreement, I would say, about doing it well, I guess. Moving beyond ACEs. ACEs are great. I've done work in acute stress manipulations where I've kind of brought people in and you can say, okay, what's something really stressful someone can do? Well, they can give a talk in public. Very reliably increases cortisol. Even worse, if you put a couple RAs in front of them and you tell the RAs, no expression on your face, don't say a dang thing, and then have them do a math task that's surprising afterwards, and have the RAs tell the participant to start over every time they make a mistake. I've had people break down on me. Like, it's a very, very stressful experience. Surprisingly, very little effect on reward behaviors or reward circuitry function. Yeah, so that's kind of part of why I kind of started to move away from that. Yeah.
Does it matter in terms of like brain development, how one consumes cannabis?
Yeah. Yeah, so I'm going to get the data. Obviously, all those things are going to matter because the time course and the amount and the concentration and the strain and all that probably matters to some extent. It's hard to know how much. And part of the problem with that is that it's been so hard to study cannabis, and legal-ish cannabis has only been around for a short period of time. So no one's really agreed on how to study it in the field. It's definitely a part where we're kind of like, okay, like, do we have standard, like, in alcohol research, we have like standard drinks and like this volume of this and this volume of that, and they're equivalent because we all know the psychoactive ingredient is alcohol. And then you talk about cannabis, you're like, okay, but actually, which cannabinoid? Well, THC, what are these other ones, and do the levels matter? And I don't know. So I think we're going to measure that, and we're probably going to have to get a lot of data on like reasons for using and subjective experiences and self-report on like duration of effects. It's going to be tricky. Yeah. 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 Yeah, puberty definitely has an effect on the brain. We're actually controlling for puberty in these studies.
Okay.
Yeah, because there are definitely effects of puberty onset. And eventually, once we have more data, we'll also have to adjust for puberty kind of rate of change, speed. Both of those are important aspects of pubertal development. And there are effects, I would say, this is kind of my soapbox, I would say in the human literature, there are not a lot of really robust sex differences in the brain, let alone pubertal effects. There are obviously some, and like men generally have larger brains, men generally have more variability in brain measurements. Beyond that, it's been pretty hard to pin down like really solid findings, I feel. And that's, in my opinion, because a lot of times we lean on kind of post hoc tests of interactions when we run out of ideas. So the literature is kind of polluted with bad results. Other questions? Yeah.
Yeah, this one. kind of, But have you like any idea, I guess, especially like, or like the environment, so like, you know, let's say, especially with like a college that you feel like more experience, like, think about like, guys, maybe you're doing 5 math at once, like.
Yeah.
Having a great, every half hour.
Yeah.
Like this, you know,
Right. Yeah. 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 The patterns and like the speed and yeah, speed of drinking and yeah. Method. I think the effects, if there are any effects of mode of delivery, I think it comes down to alcohol concentration. I think we agree that alcohol is the psychoactive substance there. So it's just alcohol concentration in terms of like beer versus liquor, whatever, what have you, White Claw. But the White Claw is dangerous because it's carbonated. So it gets you drunk really fast. The patterns of use in terms of like, yeah, not, instead of being like binge, just like an X number of drinks in 1 hour, but actually like, okay, they're like pacing themselves, they're going hard and then backing off. Yeah, I haven't looked at that much. I think part of it is it's really hard to get that kind of data on a person's behavior in the wild.
And they're actually, it's done for privacy has, I don't know if you've seen it.
I have a lot of.
Yeah, yeah, well, yeah, and the consequence.
Yeah. Yeah, that's so that you can get some of that with the breathalyzers. I think the tricky part, even with the breathalyzer, is that it doesn't quite get the nuance in patterns that you're kind of asking about, if I understand, because it tells you about like cumulatively recent alcohol use, but it's not perfect. Yeah.
How much you use.
Yeah, exactly. That can be really good. I think the problem with the phone app, the problem with the phone app is that it creates a lot of participant burden. So you worry that people are going to stop responding, and that actually while drinking, like the high drinkers who are drinking the most, or the ones you're the most interested in, are the most drunk, are the most likely to not respond. So you're missing data is not missing at random, right? Yeah. Your buddy would be like, hey, can you go to this? I'm tired. Yeah, yeah. Oh, yeah. Yeah. Yeah, so GAP is definitely expressed in like some of those core regions. The literature is a bit biased into focusing on dopamine when it comes to reward. And the reason for that, I mean, there's a good reason for that, which is that dopamine signaling from an earlier nucleus in the pathway, the ventral tegmental area, is both necessary and sufficient to drive reward behavior. So there's a lot of reason to believe that dopamine is the key neurotransmitter, but we definitely know that other neurotransmitters play a role. And there's a lot of data mapping out those signaling pathways. Yeah. You had a question earlier on.
Yes. Oh, firstly, thank you so much for
explaining
your research so well to an audience that doesn't necessarily have the experience.
Yeah.
I mean, like the questions you're getting, like we all understood.
Fantastic.
And so this is fabulous research. I had a question about the role of the hippocampus. Now, if you could go to the slides where you have the nice graphic from your.
Yeah, I love that picture. Where is it?
So that's great. So that means that's the causal.
Yeah, so the hippocampus is the one region where there has been repeated evidence, not in my hands, other people's hands, that there is possibly a causal effect of alcohol on lower hippocampal volume.
So is that why if you go a few slides back where you show that?
Yeah.
Regions, I think.
Other way. Here we are.
So you don't see any statistically significant.
Effects in the hippocampus. Mm-hmm.
I'm curious, like what you think is going on there, considering that it is a puzzle.
It is a causal. Yeah. Well, this is not a test of causality with the TWA. transcript one might association study called a TWAL. So it's not a test of causality. It's just a test of whether genes that are correlated with alcohol use are also correlated with natural mRNA expression in about 100 postmortem human brains.
But when do you expect there to be some gene expression happening in the physical condition? So
No, I mean, if it's purely causal, you would not expect, because you'd expect that genetic risk for use have no relationship to the hippocampus. It's the alcohol that's causing it, yeah. I don't actually necessarily believe that the hippocampus is purely causal, because at the point when we wrote the review, that was the best data. Yeah, the hippocampus, if you don't know, is a memory structure. It is really weirdly shaped. It's shaped like a seahorse, hence the name hippocampus. But it also is really weirdly shaped in a way that makes it really, really, really tricky to image well. Because it's curved, it has folds, it has multiple different subsections that are really hard to differentiate. So the hippocampal literature is a little bit more sketchy for that reason. Yeah. Yeah, of course.
then my other question, this may be off topic a little bit.
Yeah.
With these pathways that you see in alcohol, do you see overlap with binge eating disorder?
Binge eating, I don't know about binge eating. And the word binge is in both, but I don't know. Do you consider binge eating to be an addiction, or do you consider it to be kind of like a disorder related to kind of control behaviors? It's a disorder. Yes, I would not. Yes, I would not consider it. Yeah, that's kind of what I was thinking, but yeah. So so in terms of genetic correlates, all of the major substance use disorders have substantial overlap, particularly with relationship to pathways that are expressed in the brain, and the non-overlap is really like metabolic pathways that all differ, obviously, because they're all metabolized differently. I think anorexia has been done. I think anorexia loads more closely with like depression than with the addiction disorders. Yeah. Which is like anorexia is not binge eating, but it's closer. Yeah.
There was a slide where you were talking about the bidirectional effect, how alcohol before than they.
Yeah.
So I guess my question is about that
Yeah.
motivation for use, whether it's first use, or continued use.
Yeah.
Right? Are you actually reward seeking, or are you avoiding something negative? Are you avoiding
Totally.
some kind of affect?
Yeah. So I think so. The data suggests that both are at play. The associations tend to be stronger for reward seeking than for the kind of like avoidant type personality type behavior, but both are definitely present in data. A lot of the models really point to, I would say that a lot of the models, I'd say there's less known about the initiation portion because it's so hard neurobiologically to study people when they initiate use. People. Rodents is a different story. People, it's really hard to know why people start use neurobiologically, what the correlates are. Just because the timing is so hard. Behaviorally in self-report, we know that both are at play, but I think the data related to continued use has really influenced our thinking. And you raise a good point that it's probably not the whole story. And this model is like more of like a simplified model that was put out by the heads of NIAAA and NIDA as like a motivation for like thinking about them. I don't think it's the whole story. Yeah.
You think there's a divergent active development as far as like whether it's more.
But I think once AUD is developed, I think it's completely different. Then this model, and I think once the AUD is developed, I think it's really like a preoccupation and anticipating, and then this whole process starts going really fast. Binge, the withdrawal, the preoccupation. I think it kind of starts much more slowly, like they were binging for a while. And now we're going to move into an actual withdrawal for the first time. And binge and withdrawal, then eventually you'll hit 1, 2, 3, and then the cycle starts going faster.
Let's see what the thought would say. Drinking my sorrow away, right?
Yeah,
Not seeking anything.
Yeah. Yeah, I mean, things like, loneliness and like all those things, are a huge driver for sure.
Funny you bring that up. I've always wondered, like, when I was a kid, I didn't know anything about this. But whenever you think that that's how the world works and they talk about drinking all the time,
Oh, that's.
Yeah.
how that might affect. perception,
Totally.
how-
Totally.
Like,
Yeah.
It's time to go for a drink, let's start.